misuse of knowledge in genetic research. Different types of species are formed from genetic research. This can occur only within a species or between closely related species. However, scientists can now transfer genes belonging to unrelated species by processes other than usual reproduction. New species can now be formed. The need to adhere to a code of ethics in genetic research. Have you heard of Dolly? Dolly was the first mammal to be created in 1997 using nuclear transfer, in which a body cell is transferred into an egg from which the nucleus has been removed. The success raised the real possibility of human cloning, sparking an ongoing moral debate. But cloning remains an embryonic science, with only 1% of reconstructed eggs leading to live births. This is the beginning of genetic manipulation. At the end of this lesson, you should be able to understand how misuse of knowledge in the field of genetics can endanger life. Realize the importance of establishing and adhering to ethics and morals in scientific research for the benefit of mankind. Genetically modified foods have the potential to solve hunger and malnutrition problems and to help to preserve the environment by increasing yield and reducing the use of pesticides and herbicides. However, technologies for genetically modifying foods pose some risk. What are the risks of genetically modified food? Most concerns about genetically modified foods are environmental hazards and human health risks. Most practicing doctors have never seen smallpox or anthrax. Anthrax is hard to diagnose, but easy to manufacture and disperse. The military and the FBI have biodetection devices that can identify anthrax in 20 seconds. It is important to be aware of the misuse of genetic research to create horrific biological weapons. It is the responsibility of human society to determine how information of genetic research is used. Misuse of knowledge in genetic research. Genetic engineering manipulates biological toxins. For example, the botulism toxin or Botox is extracted from the bacteria Clostridium botulinum. Clostridium botulinum is a spore-forming bacteria that can grow and produce toxin in the absence of oxygen, such as in a sealed container or below the surface of food. It could be genetically manipulated, such as mutating its gene or fusing it to another molecule to stabilize it. It could be a highly infectious and toxic biological weapon. It only requires a small quantity to cause disastrous results, and it has resistance to treatment. Genetic engineering in animals raises ethical concerns about the welfare and consequences of genetically modified animals. Much harm can be caused during production of genetically modified animals. The techniques used in genetic manipulation of animals, such as laparotomy, can cause post-operative pain in order to get eggs from female animal donors. Laparotomy is a surgical operation involving an incision through the abdominal wall to gain access to any abdominal organ, like the female reproductive organs, bladder, and liver. Death can occur during development of embryo in female animals. In large farm animals, it is known that miscarriages cause distress to the mother. For research purposes, gene deletions, mutations, or defective genes may be introduced to animals to cause genetic disease. In genetic manipulation, harmful side effects occur when the introduced DNA disrupts the function of the animal's own genes. Many disruptions prove fatal to the developing embryo, producing animals with abnormalities, including loss of limbs and organ malfunctions. Ethics of genetic research. It is important to think about what is right or wrong. Codes of ethics in scientific research will make us think about the right thing to do. What are the ethics and morals in genetic research? 1. Truth. Scientists carry out their research by acting in accordance with the methods of scientific research and the rules of conduct in the scientific community in general. The scientist presents his or her data in full, precisely, frankly, and fairly. 2. Responsibility. The scientist bears full responsibility for every scientific research or experiment he or she conducts, particularly with regard to its direct effect on human lives, human physical and mental health, welfare, dignity, and liberty. 3. Professionalism. The scientist engages in his or her scientific pursuits in a wholly professional manner, making judicious and continual use of the special knowledge particular to his or her area of expertise. The scientist imparts the values and principles of scientific research to all those conducting research or experimentation under his or her supervision. 4. Freedom. Scientist serves the goals of scientific research based on the principle of scientific research freedom. The scientist is willing to undertake the obligation to comply with practical restrictions in the areas of development and application to the extent that these are required by social or economic considerations in keeping with the principles of the democratic system. 5. Collaboration. The scientist acts within a universal framework based on scientific goals. This can be done by maintaining openness, mutual assistance, and trust. The scientist performs every scientific act in accordance with all of the requirements of the scientific method and at the highest standards. The need to adhere to a code of ethics in genetic research. Click the box to know what you have learned. Genetic research. Misuse of knowledge. Biological weapons. Abnormality in animals. Consequences. Environmental hazards. Human health risks. Ethics and morals. Truth, responsibility, professionalism, freedom, collaboration.
Debate on Code of Ethics in Genetic Research. When Ian Wilmot of Edinburgh's Roslin Institute announced the successful cloning of an adult sheep in 1997, it was the most significant breakthrough of the decade in the scientific world. The success raised the real possibility of human cloning, sparking an ongoing moral debate. Should we try to change or add genes in human cells? Analyze the issue of human cloning and identify the ethical issues they present. Genetic engineers could eliminate defective genes, change existing one, or even add a few extra. Once a genetic engineer has changed the genetic composition of a human egg fertilized in a lab dish, the egg divides over and over to form all the tissues of the body to clone a human. Ethical issues of human cloning. We have to distinguish between cloning of cells for medical uses on a patient and an entire cloned baby. Cloned tissues could be used for transplants and would cut down on animal experiments. But most people see the cloning of a new human as unacceptable, as it is an offense against human dignity and that each individual has a right to his or her own genetic identity. In fact, there is already a market for pet clones. But do animals have a right to their genetic identity? What are the advantages of genetic research? 1. Increase yield of crops. 2. Create biological weapons. 3. Produce medicine to treat genetic disease. 4. Ensure the continuation of endangered species.
Your answer is correct.
What are the potential risks of genetically modified food? 1. It increases the diversity of plants. 2. It reduces effectiveness of pesticides. 3. It has potential risk to harm other organisms. 4. It triggers allergic reaction in susceptible individuals.
Your answer is correct.
Which of the following is not a misuse of genetic research? 1. Creating biological weapons. 2. Reducing the use of pesticides. 3. Producing genetically modified food. 4. Causing the death of the embryo in female animals.
Your answer is correct.
What are the consequences of genetic manipulation in animals? 1. Causes genetic disease. 2. Is fatal to the developing embryo. 3. Producing animals with abnormalities. 4. Disrupts the function of the animal's gene.
Your answer is correct.
What are the ethics and morals in genetic research? 1. Truth. 2. Freedom. 3. Responsibility. 4. Professionalism.
Your answer is correct.